Okay, let's tackle the last objective in Unit 2. And this objective deals with describing active transport and its role in nutrient uptake from soils to plants. So, nutrient uptake, up until this point, we've described passive uptake, essentially. There's still some energy because the plant root needs to grow and it needs to develop root hairs and it needs to develop lateral roots, and that all requires energy. But other nutrients are still taken up, and it's not just passive uptake, it's active uptake. So, let me go back. So, humans are active, we're expending energy. Plants do the same thing. They do the same thing. Okay, so plants are doing the same thing as when we're out running or biking or kayaking or whatever we're doing. Plants are doing the same thing. They are actively expending energy. And so ion uptake by plants is characterized by a number of different factors. And we're going to talk about these, and we're going to tie this in with what we just talked about in the previous video. So, uptake of ions by plants is characterized by selectivity, that certain elements are taken up preferentially while others are simply discriminated against or even excluded, by accumulation. Oftentimes, and in fact, this happens almost all the time, the concentration of elements can be way higher in the cell sap than in the external solution. All right, so plants are fighting against a gradient. They're fighting against keeping the nutrients high in the plant itself and fighting against them being re-released back into the soil solution or another part of the plant altogether. They're fighting accumulation or a concentration gradient. They're fighting against different genotypes that are present within plant species themselves. So different plant species operate differently in terms of accumulation and selectivity. And so how does this all happen? How can this all be passive? It really can't. It cannot be all passive. It cannot be all driven by root interception, mass flow, and diffusion. It just can't. So up until this point, we've talked about passive uptake, but active uptake is what you see in this picture. And active uptake ties directly in to the points made on the previous slide. Active transport or uptake simply moves solutes against a concentration or electrochemical gradient and requires the input of energy. And it also typically requires hydrolysis or the splitting of water into hydrogen and hydroxide in conjunction with conversion from adenosine triphosphate energy, right? It's an energy molecule to ADP and the release of phosphate and energy. Okay. And maybe you've learned about some of this in some of your previous classes, but what you see here is active transport of sucrose. And this would be active transport of sucrose to say inside the cortex of a root, where the concentration of sucrose or carbohydrate is relatively high, and the concentration outside of the cortex cells is relatively low. And how do plants store sugars or carbohydrates in those cortex cells? They do it against the concentration gradient. So how do they do it? Well, inside the cell itself, there's the creation of free protons in the form of hydrogen. This is formed by the splitting of water, hydrolysis, the splitting of water. And then to that requires energy. And then to pump this hydrogen across the plasma membrane requires energy in the form of ATP, which pumps hydrogens out to the outside of the cell. There's a gradient that's generated where the concentration of hydrogens outside of the cell are greater than inside of the cell. And there's a transport mechanism by which those hydrogens can flow back in. And when they flow back in, this transport mechanism can also carry sucrose to the inside of, say, the cortex cell. This is all active transport because it requires a great deal of energy in the form of ATP. So we've learned about uniporter or uniport mechanisms. The uniport in this picture is one particular solute or ion that's moved directly across the membrane in one direction. And this is facilitated diffusion or passive. This is passive, okay? And this mechanism is what we described, and the word diffusion makes this a little confusing, but this uniport, where you have passive uptake, describes nutrients that are taken up via root interception, mass flow, or diffusion. Then you have a symport mechanism and an antiport mechanism that require active uptake, requires ATP. The symport is two different solutes move simultaneously across the plasma membrane in the same direction. We just described this on the previous slide with the accumulation of carbohydrates or sucrose in cortex cells. The opposite occurs in antiport, where one ion is pumped out from the inside of a cell and another ion or compound is pumped into the cell. So they move in opposite directions. So let me give you an example of both of these. I know we already talked about symport mechanisms, but let's talk about these both again. So this mechanism on the right is symport, and we just talked about this. This is where free hydrogens are pumped across the plasma membrane and they flow back in through a transport mechanism that also carries sucrose or glucose or carbohydrate into a cell, like a cortex cell. The antiport is a little bit different, and the antiport is something that could happen on the edge of a root cell or root hair itself, where the plant wants to take up potassium, but in order to do so, it has to pump something else back out, like sodium. For some reason, the plant may have taken up sodium for an ion balance to maintain ion balance inside the cell, but it doesn't need sodium, so it pumps it back across via the use of ATP, and then potassium flows back in. Okay, so symport is two ions or compounds moving in tandem across the plasma membrane, and antiport is one is moving out and one is moving in. I hope that makes sense. And that brings us to the end of Unit 2, Soil-Plant Relationships. Good job.